phenotyping. Today we have 4 speakers, and after that we have some time for discussion. Unfortunately, the time is restricted to 2 hours for the workshop, so after inviting the speakers, I realized that there's maybe not too much time for discussion left, but we'll see how that works. Therefore, I want to give you a very, very brief introduction into some work we did in our institute. And I want to... maybe I come back to this picture. This was from last year. One of the first workshop we did in Jülich with a small group of participants. And we like to continue this series for the ongoing years. About the activities I reported already of the working group, I reported already in the assembly, but maybe not everybody was there, so I repeat this very briefly. We had 3 workshops in the last year, including this one today. The other one was the one which we had in Ghana, where we introduced our mobile application that we developed in-house, that I will also present in the next slides very briefly. We introduced that to the farmers or interested people there in Ghana who participated in the workshop in order to facilitate their work, in order to monitor growth in plants. And the second workshop was in Thailand. This was not focused on a specific app or development, but rather on how to use public or open source software to work on different imaging problems. In this case, it was in rice, and in rice which was damaged or infected by some diseases, how to extract the disease levels from images using a software like ImageJ and Fiji. Today, I will give you a short introduction into our smartphone app, which we developed. After that, we have our first main speaker, David, who will report on coast-to-coast low-cost plant imaging. After that, we will have a very brief talk by Paulo Hermann on low-cost and disposable sensors. And lastly, Noah Felgren will report or will talk about Raspberry Pi powered imaging and open source software for plant phenotyping. And finally, we'll have our general discussion. I checked the website plantimageanalysis.org for the existing apps regarding estimation of leaf area or canopy leaf area. This is a very good website to have an overview of the existing solutions, and I found quite a few which are related to leaf area or things to canopy or to leaves, not only leaf area but also leaf diseases and leaf identification and so on. We were particularly interested in monitoring leaf area non-invasively, and I found 3 apps which are quite useful, especially the first ones under very controlled conditions, which means usually you have to detach your leaf and image that with a suitable background, and then you get very suitable results, maybe also together with calibration target, which gives you then also results in metric measures. And I found one which is called Easy Leaf Area. Sorry, I'm switching on your presentation, so you have to switch back later. I use this button. Easy Leaf Area is an app which also already uses RGB channels in order to distinguish between leaf and background, and they also applied this under non-controlled conditions in the field, like you can see on the images. Besides a very good, I think it's a very good app, and it's applicable outdoors. And it also has this calibration target to get real measures. But still, the problem with these apps is that they are not very flexible and maybe tuned to specific conditions. So our aim was to develop an app which is more flexible and applicable in different imaging situations and also with different targets, not only green leaves, but also other things, which I will show you in the next image. So here is just an overview over images that we segmented with the app. So on the first row you see, for instance, 2 plants from a study in with teff, and the other one is banana, and that worked pretty well because we did it under controlled conditions in the imaging chamber. But it also worked pretty well on roots. Here in this example we have cassava roots in different growth or developmental stages, which we tried to segment in order to estimate the root, proxy for root mass. And we also tested it on different flowers and on seeds, and partly with a natural background, partly also with artificial backgrounds, like in the seeds with these blue sheets. And it worked pretty well. And we also tested it. First, let's have a brief look at the app. Here you see an image which was taken and segmented. So we introduced several methods which can be easily parameterized, and I want to go into detail here a little bit, showing the approaches which we had. There's one. So we implemented different segmentation approaches to come to a good result, which can be used depending on imaging situations and so on, or also depending on the processing time which one is willing to use or to have. One is based on single channel segmentation, the other one is on greenness indices, so indices which are computed from RGB channels, and the last one, which is pretty powerful and frequently used also out in other applications, HSV segmentations, so in a converted color space on all channels in combination. And we also included calibration, of course, and we also included some generic algorithm part in order to optimize these segmentation parameters. This, of course, requires training data, and this can be done on the app if you like, but also outside, of course, and then you can use these parameters for your data set to make, for instance, batch segmentation on a larger data set. I want to come to, I think it's already my last slide. Here we tested our approach against one of our established systems, which is called Screenhouse. So this is one of our Greenhouse systems. Here we use support vector machine classification for distinguishing between foreground and background. So what you see on the diagrams on the left side are the results from the greenhouse, and we plotted the leaf area, the one which we measured with a leaf area meter, so from destructive measurements, against the leaf area which we estimated with our app. We did this for both greenhouse and our mobile app, and you already see that the results are pretty similar, which gave us a good hint that our solution works. So in these controlled scenarios, it's of course not a big, it can be a problem, but we hope also that these approaches work outside under field conditions. There we did not have any test measurements so far, but this gives us a good hint that it's working pretty well. And with that, I already want to end my introduction with more credits regarding our group, which was involved in this project. And thank you for your attention. And if you have any questions, I'm happy to answer them.